Esteemed Nobel laureates, dear colleagues, friends, and all of you who have joined us here today in Aula Medica. On behalf of the Nobel Assembly at the Karolinska Institute, it's my great pleasure to introduce this year's Nobel laureates, Dr. Shimon Sakaguchi, Dr. Mary Brunkow, and Dr. Fred Ramsdell. The three laureates have been awarded the 2025 Nobel Prize in Physiology or Medicine for their discoveries concerning peripheral immune tolerance. The Nobel Lectures are the start and the scientific highlight of the Nobel Week. The findings about which we are about to hear are fundamental to our understanding about how immune defenses are kept in check. The identification of molecularly defined regulatory T cells launched an expansive research field of relevance to many areas of medicine: autoimmunity, organ transplantation, cancer biology, and neuroimmunology. We live in a world full of microorganisms, and we depend on them to maintain a healthy ecosystem on our planet and inside our bodies. To protect us from potentially disease-causing infections, we have evolved an amazing immune system that has equipped us with highly effective layers of control. Immune responses are powerful and they must be regulated. To fine-tune these strong responses, many feedback regulation mechanisms exist. While all living organisms have evolved some sort of defenses, the adaptive immune response is particular to vertebrates. Central to adaptive immune system are T and B cells, each of which express a unique antigen receptor on its surface. These receptors are generated through combinatorial gene rearrangements, providing us with an extremely diverse repertoire of receptors that mediate recognition of all potential intruders that might infect us. But how can the adaptive immune system distinguish between foreign structures and our own tissues? How is immune tolerance achieved? This question has puzzled immunologists for a long time, and one critical answer was provided with the description of central tolerance, a process whereby self-reactive T cells are deleted in the thymus, preventing potentially harmful cells from entering circulations. But this did not explain everything, since some self-reactive cells are known to escape thymic selection. Many groups were interested in understanding the mechanisms behind immune regulation. But the experimental toolbox was quite limited during the 1960s and 70s, and although it gradually improved, it was nothing like today's easy access to monoclonal antibodies and single-cell sequencing methods and other tools that greatly help today's researchers in defining cellular subpopulations. The discoveries for which the 2025 Nobel Prize is awarded is a story of two research fields coming together. The first involved findings in cellular immunology, and the second in molecular genetics. When the pieces were put together, the findings resulted in the definition of regulatory T cells, cells that really stand out among other immune cells, since instead of attacking, they dampen immune reactions. The story starts with Dr. Sakaguchi, who, after completing his medical studies at Kyoto University, was determined to follow up on previous observations that removal of the thymus from 3-day-old mice resulted in severe autoimmunity. By performing a series of cell fractionation and transfer experiments, he could show that CD4 T cells that also express the cell surface marker CD25 display regulatory functions. The results were published in a landmark paper in 1995. Much earlier, in the 1940s, at the Oak Ridge National Laboratory in Tennessee, researchers had noticed a spontaneous mutant mouse strain, which they named scurfy. The male scurfy mice developed severe autoimmunity, while the female mice did not. This suggested that the mutation was located on the X chromosome. This mouse intrigued the personnel to such an extent that the scurfy mice were kept going for half a century. This in itself perhaps deserves a prize. They realize that this may be of great scientific value one day. But it was not until the 1990s that the scurfy mice finally were subject to detailed molecular and genetic characterization by Dr. Brunkow and Dr. Ramsdell, who at the time were active at the company Celltech Karlsson. The severe autoimmune phenotype that the scurfy mice displayed suggested to them that if they could only find the mutation, this might provide really crucial clues to understanding immune regulation. As so often when great discoveries are made, it's about identifying an important question and pursuing it. As we will soon hear from the laureates themselves, this work led to the identification of FOXP3, a gene that encodes a transcription factor that programs T cells into becoming regulatory T cells. Soon after the discovery of FOXP3, Dr. Brunkow and Dr. Ramsdell showed that young boys with the severe autoimmune disorder IPEX also had a mutation in the FOXP3 gene, demonstrating its relevance to human disease. These landmark papers were published in 2001. Following these discoveries, Sakaguchi's team connected the two findings when he showed that FOXP3 was selectively expressed in CD4 and CD25 positive T cells, while Ramsdell team showed that the regulatory T cells were absent in the scurfy mice. With these pivotal results, the floodgates opened and immunologists all over the world began to study regulatory T cells. The aim was, of course, to understand their biology and to develop clinical applications. On behalf of the Nobel Assembly, it's my great pleasure to introduce Dr. Shimon Sakaguchi, who will be our first speaker this afternoon. Shimon Sakaguchi was born in Nagahama, Japan, and he received his medical degree from Kyoto University's medical school in 1976. He then received his PhD from the same university in 1983. Following some years as a medical resident and about 10 years as a researcher in the US, he developed this field. He was active both at the Johns Hopkins University, also as a visiting scientist at Stanford University, and the last 2 years of his US tenure he was active at the Scripps Research Institute in La Jolla. After returning to Japan in 1991, he worked as an investigator for the Japan Science and Technology Agency, and he was a professor at the Department of Pathology at the Institute for Frontier Medical Sciences at Kyoto University, where he was also the director between 2007 and 2011. Since 2011, he is a professor at the Department of Experimental Immunology at Osaka University, where he's also the vice director since 2012. Please join me in welcoming Dr. Sakaguchi to the stage to deliver the first Nobel Lecture in Physiology and Medicine 2025.
Thank you, Professor Hedestam, for a kind introduction. So, Excellencies, ladies and gentlemen, it is my great honor and pleasure to receive this year Nobel Prize in Physiology or Medicine, together with Mary Brunkow and Fred Ramsdell. I'm grateful to the Nobel Committee and Nobel Assembly at the Karolinska Institute for selecting us for the award. So today, I'd like to talk about the role of regulatory T cells in immune tolerance. As shown on this slide, the immune system, it protects our body from invading microbes. It has unique properties. T and B cells, they have a reactivity to diverse array of antigens. They have a high antigen specificity and immunological memory, and also a potent mechanism of antigen elimination by the antibodies and the T cells, helper T cells and killer T cells. And also the immune system, normally it doesn't react with self-antigens. At the turn of the century, Paul Ehrlich, he coined the term the horror autotoxics. It means the horror of self-toxicity. He thought that normally the immune system doesn't react with our body. But at the same time, he stated that there must be certain regulatory contrivances which protect ourselves from autoimmunity. Since then, several mechanisms of immune tolerance have been proposed. One is deletion. So immature T cells or B cells, when they recognize self-antigen, they may be deleted, kind of apoptosis, deleted from the immune system. Second idea is inactivation. So usually T cell need not only antigen recognition, but also co-stimulation. Just on recognizing antigen, then these lymphocytes become inactivated. In immunology terms, anergy. And third mechanism is suppression. Even normal individuals, they may possess a potentially hazardous self-reactive lymphocyte, but their activation and expansion is controlled by another T cell population. Now we call them regulatory T cells. So the key question is, which mechanism, well, if it doesn't work, whether it can cause autoimmune disease or not. So autoimmune diseases, it is estimated that approximately 8% of the population is afflicted with some sort of autoimmune disease. For example, multiple sclerosis in the nervous system, thyroiditis or gastritis, or the adrenal gland, or Langerhans islets in the pancreas. All these, the autoimmunity in these organs, it's a very popular autoimmune disease in humans. And then sometimes ovaries or testes, they are also affected, causing infertility in female and male. And also, there are systemic autoimmune diseases, such as SLE, systemic lupus erythematosus, and rheumatoid arthritis. So, and then one feature of these autoimmune diseases, that is overlapping. So multiple organs are frequently affected. For example, type 1 diabetes, thyroiditis, and then gastritis. So from that point of view, there is an interesting experimental system developed by two Japanese pathologists, Yasuaki Nishizuka and Teruo Sakakura. They found that when you remove the thymus around day 3 after birth, this neonatal thymectomy can cause various autoimmune diseases. For example, here, strain A, B, C, strain A, when they are removed of the thymus around day 3 after birth, the 30% developed the autoimmune gastritis, and around 30% autoimmune ovulitis, autoimmune disease in the ovaries. But interestingly, day 0 thymectomy, thymectomy during 24 hours after birth, almost nothing happened. The day 7 thymoectomy, also nothing happened. If you change the strain to strain B, and now the day 3 thymoectomy caused the ophlitis, about 90%, with some other autoimmune diseases. In contrast, strain C, if you remove the thymus around day 3, nothing happened. So this means that the day of thymoectomy is critical. And also, if you change the genetic background of the mice, you see other autoimmune diseases with different frequencies or resistance to autoimmunity. So how neonatal thymoectomy should cause autoimmune disease? So what we found is that if you inoculate normal T cells from adult mice, especially CD4 T cells, after neonatal thymoectomy, then you can prevent disease development. So how? Our hypothesis was the thymus is producing T cells from gestational days on. So day 0, mice were born. So day 3 thymoectomy, it means perhaps before thymoectomy, perhaps the thymus has already produced the autoimmune T cells. And then around day 3, they start to generate the kind of autoimmune suppressive T cells. Now we call them regulatory T cells. So day 0 thymoectomy, no T cells, no autoimmunity. Day 7 thymoectomy, already enough number of regulatory T cells in the periphery, so no autoimmunity. Now the question is, how we can separate these two populations, regulatory T cells and autoimmune T cells, from normal adult mice? And then whether if we remove regulatory T cells directly from normal adult mice, can we induce autoimmune disease or not? And so here, normal mice in the spleen, lots of T cells, and then we started to dissect the T cell population in this in splenic T cells by the expression of various cell surface markers. So how to remove this? It's an antibody plus complement, and then the antibody react with this certain molecule, these molecules, and then they kill these cells. And then after that treatment, if you transfer to the T cell deficient mice, and then if you see the autoimmune disease, then this means that removed population may be suppressive and then inhibiting the autoimmune disease development. So first we are successful with a CD5 molecule. And then 5 years later, Fiona Powley and Dan Mason at Oxford University in the UK, they showed that CD40RB is another marker for that population. And the question was then, by removing the smaller number of T cells, and then if we have the broader or more serious autoimmune diseases, that is a more specific marker for this population. So, and then we end up with the CD25. So here, this is a flow cytometry, and each dot means each lymphocyte. So here, CD25 positive and CD4 positive. This population, when it is removed by antibody, anti-CD25 antibody plus complement, and then this population is gone. And then if you transfer to T cell deficient mice. So now you see various autoimmune diseases. So for T cell to transfer to T cell deficient mice, nothing happened. And then if you transfer CD25 minus cells, 100% of mice developed gastritis, autoimmune disease in the stomach, ovaries, ophlitis, 100%, thyroiditis, 70%, and then also in other organs, salivary gland, adrenal gland, islet, and type 1 diabetes, kidney, glomerulonephritis, as seen in SLE, or joint the arthritis, as seen in human rheumatoid arthritis. And importantly, if you add back the removed population, CD25 positive cells, now you can suppress disease development. So in vitro, we can demonstrate their suppressive activity. So in the spleen, CD25 positive cells, around 10% of CD4 positive cells. And also in the thymus, the 5 to 10% of CD4 positive cells. And then they are not, if you remove them, CD25 positive cells, now you can enhance the proliferation of responder T cells, CD25 minus cells. And more importantly, if you co-culture with CD25 positive Treg here, and then responder T cells, now Treg suppress the proliferation of this population in a dose-dependent fashion. And so Treg also suppress CD8 positive cells. Similarly, in the thymus, CD25 positive here, regulatory T cells can suppress the proliferation of other lymphocytes. So these findings suggest that the thymus produces Tregs as a functionally distinct and the competent T cell population. So here in the thymus, the Treg differentiate into mature population. So compared with other T cells, it may take longer for them to become mature within the thymus. That is the reason that their differentiation or generation is a bit delayed. And so the neonatal thymectomy around day 3, this means that by day 3 thymectomy, a bit the Treg generation is delayed. So you can selectively or relatively reduce Treg, and you can cause autoimmune disease. And second, thymic Treg population the links central and peripheral tolerance. So the T cells react with the ubiquitous or abundant self-antigen, and they'll be deleted by a clonal negative selection or clonal deletion. And some of them with intermediate affinity for self-antigens, somehow they differentiate into Treg. And then low affinity ones, they can escape the negative selection and go to the periphery. And some of them self-reactive T cells, when you remove Treg, they become activated and cause autoimmune disease. So central tolerance and the peripheral tolerance, somehow they can be connected. And then also, whatever it is, genetically or environmentally induced alteration in Treg number or function can be causative of autoimmune disease. So we have shown that the various environmental causes, for example, certain virus or certain chemical or the X irradiation, all can reduce regulatory T cells and then cause autoimmune disease. Like the cancer development, the viral carcinogenesis or chemical or the physical carcinogenesis, just the affecting the oncogene or tumor suppressor genes. So, but in autoimmune disease, not gene, at the cellular level, this population is affected, and you see autoimmune disease. And then various molecules, if you look up the knockout mice, they develop autoimmune disease. Now the question is whether this kind of mechanism is also operating in humans or not. And then the very interesting findings were made by the Mary Brunkow and Fred Ramsdell in 2001. So they positionally cloned the genes responsible for scurfy mouse, very severe systemic autoimmune disease. And then immediately they showed that this gene, they named FOXP3, a transcription factor. It is responsible for human rare autoimmune disease, IPEX syndrome. So IPEX syndrome, the one gene defect can cause the autoimmune disease in various organs, and especially type 1 diabetes and inflammatory bowel disease. And so after when milk feeding starts, and then intestinal microbes develop, the commensal microbe develop, and then you see the start of inflammatory bowel disease. And allergy, when the baby starts to take food, immediately they show food allergy. So we got interested in this gene because the disease picture is so similar to what we saw after Treg depletion. So later on, we made an antibody against FOXP3, And then we found that normally 10% of CD4 T cells express FOXP3, but in SCARF mice, no such cells. And now we can stain regulatory T cells with FOXP3 and then CD25. So what we found is that the majority of FOXP3 positive cells, they are CD25 positive in the spleen, also in the thymus. So mouse study and the human IPIC syndrome, it can be connected. So 3 groups, including us, and then the Sasha Rudinsky's group in Seattle, and the Fred Ramsdell group in Seattle, all 3 groups published this finding that FOXP3 is specifically expressed by regulatory T cells. So what does this mean? Here, the potentially hazardous T lymphocytes are present in every healthy individual, but under dominant suppression by Treg. So it's amazing that the FOXP3 mutation, the 80 to 90% of kids, the affected patients, they develop type 1 diabetes. This means that whatever HLA they might have, the Treg deletion or Treg defect can cause the type 1 diabetes and other autoimmune diseases. So, and then second, the border between self and non-self is not fixed, but movable by controlling the number and function of Treg. So self, non-self discrimination, the immunologists that prefer that word, but a border between self and non-self, it is not fixed. You can move this by the increasing Treg or reducing Treg. So this means that autoimmune disease can be treated or tumor immunity evoked and transplantation tolerance established by controlling the number and function of Treg. So this is the tumor immunity. Here, if you reduce the number of Treg, and then this is a T cell deficient mice transferred with CD20, the 4 T cells or Treg depleted cell population. And if you put in the inoculate tumor cells, without Treg depletion, a tumor become bigger and bigger, and then all the mice died of tumor progression. But here, without Treg, first tumor become bigger and then rejected. And after rejection, if you immunize with 10 times the number of tumor cells, they are immediately rejected. So this means that, like a measles infection in viral infection, we can make the normal mice specifically immune to the tumor cells. How about transplantation? If you raise this border up, and then you can induce transplantation tolerance. So again, T cell deficient mice. So without T cells, they can accept the skin graft from other strains of mice. And then if you inoculate, so 100% accepted. And if you transfer the normal T cells, strain A, and then you see the rejection happens. So within a month, all the grafts rejected. But if you first transfer the Treg, 1 week before, then Treg are recruited to the graft site and they expand, and then suppress the rejection. So the rejection become delayed, and 30% become permanently tolerant. And if you increase the number of Treg, now more than 70% become permanently tolerant. So without using immunosuppressive drug, just expanding naturally present Treg, we can induce transplantation tolerance. So with the discovery of CD25 as a marker, cell surface marker for Treg, and FOXP3 as a specific marker in the nucleus, now we can define Treg as distinct cellular entity. And then publication on Treg now rapidly increased, and then especially it allowed or enabled the deciphering the molecular basis of Treg development and function. And also, it has enabled clinical application of Tregs. So what's the molecular basis of the Treg function? So here, the Shohei Hori in my lab at that time, so he expressed the FOXP3 gene in normal T cells by retroviral vector. And then without Treg, and you transfer Treg depleted cells into T cell deficient mice, you'll see the severe autoimmune gastritis and colitis, IBD, inflammatory bowel disease. But if you co-transfer with FOXP3 transduced normal T cells, now they can suppress the disease development. So FOXP3 gene can confer a suppressive activity on normal T cells. So how? FOXP3 controls hundreds of genes, either activate or depress. For example, CD25 become activated, CTLA4 activated, IL2 interferon gamma depressed. The question is, among the FOXP3 control genes, which one is important for this suppressive activity? So this means that which gene, when it is become deficient, whether such mice develop the severe autoimmune disease as seen in FOXP3 mutation as scurvy mice. There are not so many. One group is IL2 and the IL2 receptor. So CD25 is the IL2 receptor alpha chain. And then another one is CTLA4. So one cardinal feature of Treg, that is they are constitutively expressing the high affinity IL2 receptor composed of alpha, beta, gamma. Already, when they are produced in the thymus, they are already expressing the high affinity IL2 receptor. And FOXP3 repress IL2 production. So normal T cells, when they are activated, they express the IL2 receptor and also produce IL2. And then IL2 bind to the receptor, and then T cells become, they expand. But on Treg, they don't produce IL2. That is the cardinal feature of Treg. So it means that they are highly dependent on exogenous IL2 for survival. So if you neutralize IL2, just injecting anti-IL2 neutralizing antibody, then immediately you see the reduction of the Treg in the spleen, also in the thymus. And then you'll see the very severe autoimmune diseases. So in this case, it's an NOD mice, the type 1 diabetes prone, genetically prone, and then the diabetes become the more severe. And then thyroiditis, the thyroid is almost completely destroyed, and the salivary gland also destroyed. And then how about the CTLA4? So Kajsa Wing, she was a postdoc from Sweden, and she showed that when you make the CTLA4 Treg specific and CTLA4 deficient mice, conditional knockout mice. So here, FOXP3 positive cells, they are expressing CTLA4. And then if you make a conditional knockout mice, FOXP3 positive cells here, but then they are not expressing CTLA4. And these mice spontaneously developed very severe autoimmune diseases, such as autoimmune myocarditis. And they show the infiltration to the cardiac muscle, and then the cardiomegaly, and then they die by heart failure. And if you look at the IgE in the circulation, the concentration is several hundred-fold higher, meaning that they are prone to develop the allergy. And then CTLA4, as you may know, anti-CTLA4 antibody now under clinical use, ipilimumab. So, these mice die soon. So we transfer the spleen cells from these mice to normal T-cell deficient mice, and then inoculated tumor cells. And then red lines, conditional knockout mice, they rejected the tumors. But in control mice, all of them died of tumor progression. So just the CTLA4 deficiency in Treg, it is very similar to what we see after removing Treg. So CTLA4 is the important molecule for Treg function. So this is our current understanding of how the various molecules expressed by Treg, how they are involved in immune suppression. So here, CD80, CTLA4 interact with CD8086. So they are called co-stimulatory molecule. For responder T cells, antigen recognition plus co-stimulation, then they become activated and expand. So if Treg reduce the expression of CD8086 by trogocytosis and then transendocytosis, so the just snatch out the CD8086 from the cell surface of antigen presenting cells. And then also the soluble CTLA4, they block the residual CD8086. So very efficiently, the Treg block the co-stimulatory signal. And second, the PD-L1, which is again, it's a good target for cancer immunotherapy. So PD-L1 is normally associated with CD80. So CD80 become reduced. Now free PD-L1 increase and then interact with PD-1 positive T cells. Namely, Treg can suppress activated PD-1 positive T cells. And if the Treg further differentiate, they also secrete immunosuppressive cytokines, IL-10 or TGF-beta. And one of the key targets is antigen-presenting cells, their expression of co-stimulatory molecules and MSC. Plus, the high-affinity IL-2 receptor, the Treg, they absorb IL-2 from the surroundings and then depriving IL-2 from responder T cells. So with these findings that the various molecules, key molecules are now expressed. And then the next question is the autoimmune disease of, the monogenic autoimmune diseases, FOXP3, CD25, CTLA4, all develop very severe autoimmunity. And now they are called T-regopathy, and then list of the T-regopathy is now expanding. And how about the autoimmune diseases, general, polygenic one, which afflict around 8% of the population. So we know that the single nucleotide the polymorphism, SNP, it can contribute to the genetic disease susceptibility. So we analyzed the autoimmune SNPs. So they are expressed by many of them, 60% expressed by the immune cells, especially CD4 T cells. And then the question was whether this variation is the loss of function effect on Treg or gain of function for helper or other CD4 T cells. And the conclusion was many of them are controlling the Treg development. And so the enhancer region of Treg signature genes, lots of Treg, the autoimmune SNPs, and they locate and then contribute to genetic susceptibility to autoimmune disease. Now, how about treatment? So you can expand Treg because they are they are expressing the high affinity IL-2 receptor constitutively. So just low dose IL-2 can selectively expand Treg. It's not enough for expansion of other T cells. So you can expand Treg and then strengthen immune tolerance. And second approach is the Treg they are present in the circulation. So take them out and then expand them and then transfer back to the patient. The third one is this is what we are working on. How we can convert antigen-specific T cells into Treg in vivo or in vitro. And so the effector cells, they are converted to Treg, FOXP3 expression plus Treg-specific epigenetic changes together. And now we can have a very stable Treg. And if you transfer back to the patient, you can suppress disease development. So now Tregs are ready to go to the clinic. So as I told you, by expanding Treg or strengthening their function, we can treat autoimmune disease, allergy, or immunopathological diseases such as IBD and organ transplantation. And other, for example, heteromaternal tolerance to prevent abortion. Then you can expand Treg. On the other hand, by reducing their function or expanding attenuating their suppressive function, you can enhance the immune responses, such as in tumor immunity. So this is so. I'd like to acknowledge the contribution by many people. First of all, I'd like to thank my wife, Noriko Sakaguchi. She's a partner in my life and also collaborator in research. And the mentors, as I already told you, Dr. Nishizuka and Sakakura, they introduced to me how to induce autoimmunity by neonatal thymectomy. And Toshitada Takahashi, he taught me how to dissect or separate our T-cell population by antibody. And I have been collaborating with many people, including my colleagues and students and collaborators. I have mentioned some of them, but others also they contributed the progress of our Treg research. And funding source, the funding source in Japan and also in the United States. I greatly appreciate their support for our research. Thank you very much for your attention.